Yeah, I think it's. Shall we go? Okay. Good afternoon, ladies and gentlemen. A warm welcome to all of you. Welcome to the session on gene and genome editing. CRISPR could also have been in the title. There are many names out there, and we have a fantastic panel today. I think one of the best that we can actually have to discuss about that topic, and I will introduce them in a couple of minutes. But before that, let me tell you how we run the session. It's a session of 1 hour, as you all will have seen in the program, so it will end at 2 o'clock. And we will use the tool Slido. Those of you who already participated in previous sessions here in the RNI days or in any other conference, it is quite common nowadays, that tool, so you might know it. And we use that tool actually in two ways. We will use it to ask you questions in the very beginning and in the end of the session again. And most importantly, you will have the possibility through that Slido to ask questions to the panel that the panel will then later on reply to. You will also have the possibility to actually say thumbs up, thumbs down for questions that come up. So maybe we get a kind of ranking of what the joint wisdom here in the room thinks is the most important questions to ask to the panel. If you have not downloaded it yet, you can still do it. It's that app, that conference app B2Match. If you download it, then in there you will have these functions. And if you're not really keen on it, don't worry, we have still also the classical microphones that we can use at the end. So that's also still there as an emergency solution. Now, gene or genome editing has been called the biggest biotech discovery of the century. I think it was the MIT Tech Review or whatever that newspaper journal is called, who has said that. And I'm almost sure that everyone here in the room has heard about it under the one or the other name. And many journals have written about it. Also, daily newspapers have written about it. It is something that many researchers really are fascinated by and really see a huge potential. And we will hear from the panel about that. Now, we also heard, and you will have heard about it and been seen writing about it, that actually slowly that new technique, which has only been discovered in 2012, developed in 2012 for the first time, it moves also already in something which seems to be applications that go outside of the research field. You will have, many of you will have heard, maybe all of you will have heard about those Chinese twins, Lulu and Nana, who were reportedly, I am personally still not 100% certain whether it is correct because, well, I'm not sufficiently into it, but who reportedly have been modified before birth, genetically modified, so to avoid that they would get later on in their life HIV. Now, this and other elements have triggered also that some researchers asked for a moratorium and they wanted a debate taking place and on germline editing, which is one specific aspect of gene editing, and they wanted also an international framework, governance of these questions. And that gives an excellent moment now to turn to the panel, because we have in the panel, I'll introduce the panelists now. We have 3 speakers who, and at least 2 of them are probably able to talk about this question of governance and framework. And we have here, if I start with Robin Lovell Badge, he is a member of a WHO, World Health Organization expert group that is currently working on questions of governance and oversight of gene editing. Robin has in the past worked also with the US American Academies of Sciences and is still working with them, and probably has also quite some insights in what thoughts are on the other side of the Atlantic. And I think you were actually chairing the panel in Hong Kong when that announcement was made about the
I was indeed, yes.
about the twins.
That was,
so maybe we'll hear
interesting session.
Interesting session, yes. And he's otherwise he's also a researcher, not to forget. So he's a biologist and has worked a lot on the early stages of the development of human embryos. Then we also have Christiane Wopen with us. Christiane is, I'm working with Christiane very often, and also with Janos. Christiane is the chair of the European Group on Ethics in Science and New Technologies. So in that function, she advises the European Commission. And right now, they are working in that group on an opinion on recommendations to the European Commission on gene editing. This is not out yet, so I cannot brand it. It's not there yet, but it will come by the end of the year or beginning next year, and we'll look forward to see what this will be about. I mean, I know a little bit, but it's not. That's Christiane's role to talk about it in the way she wants. She also was chair of the German Ethics Council, was a member of UNESCO Bioethics Committee. Advised also the German government and the European Commission on other aspects, not only in relation to gene editing, but also on other ethical aspects of new technologies like artificial intelligence or data governance. She is a medical doctor and a philosopher, and a professor at the University of Cologne, and there I think you focus mostly on ethical aspects of reproductive medicine, neuroethics, gene editing, quality of life, aging, many aspects. Our last panelist is Janos Bonitski. Also with Janos, I work quite a lot. He is a member of the European Commission's Group of Chief Scientific Advisors. And in 2017, and this I can actually show, he was leading work of the Group of Chief Scientific Advisors on new techniques in agricultural biotechnology, where he was actually comparing new techniques to older existing techniques. He's also a biologist and combines many different techniques, synthetic biology, structural biology, bioinformatics, in his research on proteins and RNA. And then I forgot to say who I am, actually. I notice now. I am Johannes Klumpers, and I'm not a gene editing specialist. What I do is I am heading a unit in the European Commission, and that unit is supporting the work of Janos and Christiane in their respective functions as members of those two groups. And my unit is also responsible for developing the ethical framework and the ethical screening of projects submitted to the research framework program, and also for research integrity policies. So just like you, I will be listening with great intent what our panelists will say. They will speak about for half an hour, and then the floor is yours. So the second part of the session, it will go to you and your questions. And I suggest that we start with Janos, and that Janos explains to us a little bit, because we're not totally certain whether everyone is totally familiar with actually what gene editing or genome editing is. And so Janos will start off and explaining to us what that actually is.
Okay. Thank you, Johannes. I will use my notes in order to be very precise in everything I say. So genome editing is sometimes also called genome engineering, gene editing, and other names. It's a type of genetic engineering in which the DNA is changed in a living cell. And these changes are mutations, and they can involve single base pairs up to thousands of base pairs in the DNA. The early genetic engineering techniques introduced the genetic material and mutations at random, and the gene editing techniques enable changes at specific locations. So we can mutate individual genes or individual base pairs in the DNA. And currently, the most commonly used tools for genome editing or gene editing are enzymes called nucleases, which are capable of recognizing specific sequences in the DNA and cutting them like molecular scissors. So these enzymes have a natural origin and have been adapted and often engineered to be very specific and discriminate between different sequences. And if the nuclease, the enzyme, recognizes a specific sequence in a cell where it is delivered, it catalyzes a chemical reaction generating a double-stranded break. And cells cannot live with broken DNA. Even a single break that is not repaired can lead to cell death. So there exist natural mechanisms in the cells to repair such damage, and the gene editing technology uses these natural mechanisms to introduce mutations, and this can be homologous recombination or non-homologous enjoining, two different biochemical mechanisms which lead to mutations in the site where the two strands were separated from each other and then combined again. And depending on the details of the procedure, the desired change can occur at one selected site in the genome or at more sites. And the change itself can be programmed. So we can precisely introduce a particular change of one letter in the DNA to another, or we can leave it random. So we know at which site we introduce the change, but the type of the change is not fully controlled. So many different variants can be generated. And the nucleases and the conditions of the DNA editing procedure are usually optimized for such a change to occur at only one desired target or several very precisely selected targets. But non-specific and unintended changes can arise because the nucleases can sometimes err and they recognize sequences that are not identical but very similar to the desired sequence somewhere else in the genome. And such unintended edits are called off-target effects, and they cause unpredictable effects if we don't know where they occur. And up to date, 4 different types of nucleases have been used. And one of them mentioned by Janos is based on the CRISPR-Cas9 system, where CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And it's a system that has been discovered in the nature, in bacteria and in archaea, where it is used as a natural system for defense against viruses. In the previous systems, one nuclease can recognize only one target. So for each new sequence that we want to engineer, we need to find a new nuclease. And the CRISPR-Cas9 system is very versatile because its specificity for the DNA sequence to be cleaved is programmed by the so-called guide RNA molecule, which is included. And this guide RNA is complementary to the target, about 20 base pairs. And thereby, one nuclease can be programmed or reprogrammed to recognize different sequences. And when we know the sequence of the gene we want to mutate, we can select a particular sequence that is unique. And we can program the nuclease in such a way that it minimizes the possibility of cleavage elsewhere in the genome. So for instance, to find a sequence that has very few similar sequences elsewhere in the genome. And such analysis can be programmed computationally. But of course, off-target effects can still occur. And an experimental test can be performed in a test tube, where the DNA from target types of cells can be isolated and this programmed nuclease can be used. Then we can analyze experimentally what are the sites cleaved by the molecular scissors, and both the target site and of course the potential off-target sites. And then once the actual gene editing experiment is performed in cells, we can diagnose by special sequencing experiments if any of such off-target events occur elsewhere in addition to the desired place of change. But of course, there are technical challenges, and we'll probably discuss about them in the second part of the discussion. Just a final note. I would like everybody to remember that mutation is a natural process. We are all mutants. We are mutants, and all of you are mutants, and actually we all mutate now when we sit here. And these are random, unintended effects. And sometimes the natural mutations lead to unwanted consequences, such as genetic disease, and one of them is, of course, cancer. And we cannot really prevent such mutations from happening randomly. But this is what medicine is for, and gene editing has been proposed as a technique that could be used to reverse mutations that are unwanted. Thank you.
Thank you very much, Janos. I think we all have understood that it's awfully complicated. And depending on how good you are in biology, there is not a possibility to have just one sentence, no, to say what it is.
Yes.
It's impossible, I understand. Okay. Thank you very much, Janos. I wanted to ask Robin, why is it that really many researchers around the world are so excited about it, about genome editing?
Okay. I'm going to limit my remarks to human health, or I can go broader than that if you want. But just a little clarification from the introduction. I've actually never personally worked on early human embryos. I have colleagues who do, but it means I don't have a direct conflict of interest when I'm talking about them. Anyway. There are, we can think of broad categories of usage of the genome editing methods, simply with respect to human health. I mean, there's a huge amount of basic research that is taking place using these tools to alter genes, which are incredibly important. So they allow us to understand the role of specific genes or pathways in both normal human biology and in diseases. And diseases could be genetic diseases, or they could be infectious diseases, or adverse effects of chemicals we get in the environment even. So they're incredibly versatile tools, and their uses are very wide-ranging. So this type of research relevant to human biology makes use of human cells in culture. It can range from studies of early human development. My colleague at the Francis Crick Institute, Kathy Nayakan, is doing such experiments, trying to understand the role of specific genes that are acting in the first few days of development of human embryos. The main reason she's doing this, or long-term aims, is to improve success rates of in vitro fertilization and other assisted conception techniques, perhaps reduce the frequency of miscarriages. And of course, that's not necessarily using genome editing methods at all to do those, but it's just to get the basic understanding first, that then once you have that, you can use conventional treatments or drugs, changing culture conditions, whatever, to make things better. So there's human cells in culture. Some of these could be patient-derived human cells, so they're already carrying a particular mutation. That will include induced pluripotent stem cells, where you take, say, a skin cell, reprogram it, so that it now becomes a pluripotent stem cell, able to give rise to potentially any cell type in the body, but in the lab. So these are very valuable ways of studying genetic diseases, but also other problems that we may encounter. There's also, of course, a lot of research done on animals, so you can use these methods to create very good models for human diseases. And because the techniques are applicable broadly, you can make your animal model not just mice, which is the common one, but you can choose any particular type of animal almost, including non-human primates. So you can make very specific models for human diseases in animals that are closer to humans, and this is being done. So it follows that human cells in culture and these animal models can also be used, of course, for preclinical research, where you actually want to test potential treatments based on genome editing tools and their uses. The treatments, I'd say it's really important, as you heard, to make sure when you're doing these preclinical tests to try and you've got established methods that are most efficient, but have the lowest frequency of off-target events. So you want the correct on-target event and no off-target events if possible. So the treatment approaches that what people are contemplating fall broadly into two classes. So these would be clinical applications. So the somatic gene therapy. So that's where you have a patient who's suffering from a genetic disease, for example. Let's take a couple of different ones. So you could have sickle cell disease. So that's a disease of the blood system, of the red blood cells. So the idea is that you could, in this case, you could do an ex vivo genome editing. You could take out bone marrow stem cells from the patient and in vitro correct the beta globin gene back to what it should be without the sickle cell mutation, and then reintroduce the bone marrow stem cells back into the patient, where you probably have to reduce their own bone marrow stem cells to give a chance for the new ones to take over. But the idea being they take over, and so that then ultimately all the red blood cells are generated, or most of them, have a normal beta globin gene, and the patient is cured of their sickle cell disease for life from that point on. These approaches there have been attempts to do these sorts of things, by the way, previously using classical gene therapy techniques. But as mentioned, they tend to affect genes at random, so you can integrate a gene to, say, replace beta globin at random, but it's not really going to work very well. It may not be expressed at high enough levels to give you any sufficient recovery from the disease. The integration will be at random. You may affect other genes just through simply integrating into the DNA. So genome editing methods are so much more, people are really excited about them because they're very precise. You can make precise alterations in a gene, which is then going to be regulated so active correctly in the right place at the right time, the right levels.
I think.
One more.
Okay.
One more. Yeah. You've also got in vivo approaches for somatic genome editing, and that's these are more challenging, although some attempts to try and do this, because you've got to have good ways of introducing the components into cells in the body. And finally, the one that people want to always talk about is the germline, potential germline gene therapies, which will be potentially heritable changes made to DNA. There are alternatives to this that are currently being used, so we're sort of, there is some familiar territory to choosing embryos that are going to be free from a genetic disease. But in this case, you'll be actively correcting the gene within the embryo to try and get a child free from the disease. At the moment, there are relatively few patients where couples where this may be really useful for. An example would be where you have one individual homozygous for a dominant disease, like Huntington's disease, or both parents are homozygous for recessive mutations. These are fairly rare, but if techniques like somatic gene therapy and conventional treatments begin to work really well, then in a few years' time, you're going to have many more individuals who have been cured from a genetic disease in their body cells, in their somatic cells, but their germline hasn't been cured. If they want to have children, they might have to resort to germline genome editing to have healthy children. So I'll stop there.
Thank you very much. So they are excited about it because there are so many diseases which researchers believe that they can research much better using the technique, that also ultimately maybe there is a better chance of actually curing some of these difficult diseases. This is what the hope is of researchers.
Yep.
Okay. Yep. Thank you very much. Christiane, ethicists are also around the world talking and debating about gene editing. Can you describe us a little bit what that debate is about? What are the questions they look into?
Yeah, of course. So actually, from the ethical point of view, this is nothing new. The technology is new, but the ethicists discuss about gene modifying techniques in humans, plants, and animals since years and decades. But there is something new, and it's easy, though it sounded a little bit different, but in principle, it's an easy technique that can be done by a lot of people. You have not to be very specialized to do that. Yeah? Mm-hmm. So this makes it a little bit difficult because it's scaling up. Yeah? The reach is different. It's scaling up to a completely different reach, and it can be done by a lot of more people. So that's where regulation comes in. Yeah, for something that has been done before, but now can be done by a lot of more people and with a far reach. So let's then talk about different application areas. Of course, the most prominent one is the human germline intervention. And ethicists discuss about categorical and conditional arguments. So is germline intervention ethically legitimate at all, or is it violating human dignity? Yeah, because you intervene in the genome of a person who cannot agree, and it will be transmitted to further generations. So it's not only that individual that even cannot agree or consent, and it is transferred to other generations. Or are there only arguments like, is it safe enough? Do you use it for treatment? Or is it even better to enhance memory or to make it feeling no harm? So I think those objectives have to be differentiated. And the ethical debate is about, has it to be forbidden categorically? Or are there conditions where we say if they are fulfilled, even germline editing can be ethically justifiable? Then we come to the somatic gene editing. And there, of course, we have the assessment of risks and potentials. That's nothing new. But for which purpose do we use it there? So if we can cure dementia, fine. But the same thing could be used to enhance a normal memory. Is it then up to the individual to say, yes, I want to be a wonderful human being that can really have a great memory of all things that I read and all things that I have done? Is that a purpose we should say, well, that's ethically justifiable? So these kinds of questions come up in somatic gene editing. And there's another question. If you combine it, for example, with CAR T-cell therapy in oncology, that's very, very, very expensive. So what about allocation of scarce resources in healthcare systems? So who has the right then to get this kind of new therapy and innovative therapy? So justice questions come in as well. Yes. Then we have the application in the animal area, animals in the field of agriculture, animals as pets. Do we want to design pets according to the preferences of people? What about animals as food for us? So we have the potential, or the power to intervene there now with a very mighty tool. And in what kind of environment do we want to live? How do we want to treat animals? And with non-human primates, for example, there's a specific question if the 3 R's, so refinement, replacement, and what is the other one? Forgot it. Refinement, replacement, and reduction. If we need a 4th R, if this framework of regulating gene modifying techniques in animals, is that enough or not? Then we have the application in plants. This is very powerful as well. Plants for food and for feed. We know all about the difficult discussions in different societies about gene-modified organisms and the regulation and the decision of the European Court of Justice in June last year. So we see that we have to take the society with us as well. Because we are intervening in nature, and naturalness for a lot of people has a moral value. That's a very difficult topic. I cannot go into detail here. Perhaps there will be a question and we can go and we can elaborate on that. But naturalness is one of the cross-cutting issues we have to take into account there. And that's the same with gene drives. That's the last application field where one species has or is intended to be modified in a way so that we can avoid, let's say, malaria also, the transfer of malaria. And then we really intrude into biodiversity. And that's where the Anthropocene comes in, not only with climate change, but with gene editing as well, because we are shaping the environment and the spectrum of species in the way we find adequate. But what about the whole system of it, right? So I stop here, but I think that's already a spectrum of a lot of deep questions.
Thank you very much, Christiane. I think that was brilliant, and many, many, many questions have come up, and some of them are have seen through the app are relating to some of those aspects. Now, before we go to the panel question, I wanted to ask you one last question, all of you together. A difficult exercise. Imagine I were President von der Leyen. I know I have not much taken care of my feminine side today, but imagine I were President von der Leyen, and I am asking you the question, what is it for me as President of the European Commission, what is the most important things for me to know about gene editing? What shall I be concerned of?
For me, I guess it's, I think the techniques are developing very rapidly, so I have some confidence that we will end up with better ways of doing it, making it efficient and safe, hopefully. We still are lacking a lot in basic understanding of human genes and genomes, and so certainly making sure that there is good research that crosses different types of individual, from wherever they come in the world, it's really important that we have that good understanding of genes and genomes. And then generally how to promote the beneficial use of these techniques, and at the same time prevent their misuse. So we've heard that they can be misused, whether it's somatic genome techniques or what. And then, obviously they can be misused not just in the human sphere, but in animal and plant sphere. So how can we, with respect to humans, how can we stop rogue individuals or rogue clinics offering untested treatments, unsafe treatments to people? Because they will start happening if we don't think of a way of stopping them.
Christiane?
So from my point of view, it is important to see that this is a global perspective and a global dimension. That's why the WHO has the expert panel, the National Academy of Sciences and the Royal Society in the UK set up a commission in the US to deal with these topics. And what I find very important is to see that we have a very mighty tool, and it should only be in the hands of very thoughtful people who have an awareness of the ethical, the legal, the societal impact of this technique, and who are prepared to have interdisciplinary dialogues, education, by the way, as well, who are prepared to be transparent in what they do. So we have to have registries, what is going on, to be transparent to the public as well, so that everyone can learn what is going on. And I think we need deeper discussion about what the moral value of some things are that we took for granted, but which are questioned by now. So what is the moral value of naturalness? Do we want to have technical solutions for social problems? So why not acting against mass farming of animals, and instead using gene technologies to make mass farming even more possible? Yeah. So do we use it in the realm of what we are doing now, or do they point us to rethink what we are doing now? And that's the same with plants. So what is the position of the human being in the order of nature? Are we manipulating everything? Are we the masters of the universe, or are we part of nature, and so trying to use these technologies for the good, of course, but avoid to have too much risks or to change the order of nature in a way that we do not oversee with consequences of future generations.
Thank you, Christiane.
So there is a number of technical questions, very important technical questions and advances that should be addressed. I think one of the most important ones is how we can improve our ability to predict the effect of intervention, in particular in gene editing. How can we predict the effect of the gene editing event in the target site, as well as any off-target effects that may occur before the actual procedure is carried out. Being able to do this will be an exceptional technical advance, and of course research in this direction is done. But there are also, apart from the technical questions, I think we have a very important issues of understanding, and in connection to what Christiane said, I think we need a debate on what we consider as natural versus unnatural. And I'm speaking from the position of science, I would like to emphasize that I don't mean natural in normative terms of what should be, but what we actually observe to occur in the nature without any human interventions. Because there are many things that do occur in the nature without the human intervention that we consider as unnatural. And why? And second, there are many elements of our contemporary world which are unnatural, like most of the agricultural products that we eat are unnatural. They are elements of human intervention. There are basically no natural foods that we eat that we consider as natural now. So defining the concept of what is natural is very important, I think, before we move on to discussing the morality of being natural, because then there might be huge differences in who says what is natural and so on. And the final thing is, can we also speak about wanted and unwanted harmful effects when we do take into account something as a baseline? Because very frequently the discussion about potential harm of some action is carried out in the context of some idealized scenario where no harm occurs, while in reality, because of the very many random events, actually there is certain probability and frequency of harmful effects because they are natural, because they normally occur. So I think these are very important discussions concerning our understanding of what actually happens in the nature that I believe should take place at least in parallel or maybe before some of the ethical considerations that stem from the results of these considerations.
We should discuss together, actually.
Yes.
Well, brilliant. Thank you very much. We have, meanwhile, many questions. I take you have said many things. What the discussion on naturalness and the moral value of nature was very prominent, so that I certainly take from it that is a suggestion that you think is important to take. Robin was making the point that we should make use of the technique to have good research, to use to actually advance our knowledge. Yeah. That is important for you. There were many other points. Shall we have technical solutions to issues that we have created already earlier? And there. The animals, what attitude have we towards animals? This goes beyond gene editing, actually, but that enters into that picture. But we have received quite a number of questions, and I am not 100% certain whether I understand perfectly well all the colors that are underlying these and that sometimes disappear and come. So if I now decide a little bit arbitrarily which of those questions I start with, I hope you won't be angry about, because it is simply I do not know whether the questions are approved by 150 of you or only 10 or so. And I actually would like to start with one question that is pink on this thing, or what is it, violet, or whatever you call that color. And that is the one which is also an issue for lawyers and regulators, of course. How can we ensure that we make good policy on genome editing if it is so challenging for non-specialists to even understand? It is not the only technical issues on which we make policies. This is something that happens in many areas, but that is definitely one. Don't know whether you have an idea on that one.
Well, I can just start off by saying it is really, as we heard, it's really important to have proper dialogue where you have experts in the room informing in an unbiased way, which is sometimes difficult, informing others who want to learn about the methods. And this should be, there should be a very active programs of public engagement, public dialogue with members of the public, but also, of course, that has to include lawyers and ethicists and others. We are trying to do that. There have been attempts to do it already. There needs to be a lot more. It's hard to know what really works well. So actually, this whole sphere of the whole domain of how you do public engagement best to inform areas where the technology is complex is really hard to do.
Yes.
So we've done this in the past in the UK, for example, on mitochondrial replacement treatments. So that's a way to avoid mitochondrial disease. It was a very thorough piece of work, using different ways of trying to engage the public and get their opinion. But it's very expensive. So, of course, the more thorough you are, the more expensive something is. And so there has to be set aside funds for doing public engagement. I know that the EU does fund this, but it's got to be linked with particular programs of research in an active way if you really want the people to understand. So.
Thank you.
Perhaps adding up on that, there was a group at the Harvard University around Sheila Jasanoff and Ben Hurlbut, and we suggested to implement a global observatory, just to listen to those who are not listened to hitherto, because we have groups of very loud people being on Twitter and Facebook and in the public and so on, and there are a lot of people out there and we don't listen to them. Yeah, let it be almost whole continents like Africa or so, who don't have really a say in shall we change all the animals and plants and so on. Yeah, but we change a lot then when doing it there, or South Africa or South America and so on. So they're not integrated into this typically Western discussion on these topics, and I think that's not adequate when we have an issue of a global dimension. So I think this global observatory could be a good idea for public engagement and for discussion. And I think it's right. Good policy always has to rely on good information before. Yeah, but that's not specific for genome editing.
No.
So there have always to be a lot of experts from different disciplines and the public, of course, being listened to by politicians before setting up policies.
Yes, so I also agree that it's absolutely essential to improve the understanding, and with better understanding we can have better policies, and this requires better dialogue, listening to questions that have not been asked before or answered before. And we have to remember that there's different elements of the system, so science, policy, economy, values, they cannot be really separated from each other. They always come together, and but when during each debate we can try to make sure that we always know what we are talking about. So when we are discussing certain question, that we can try to make sure that we know whether we at the given moment we focus on facts or whether we focus on values and emotions, because they cannot be totally separated, but also intentions are very, 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 very important. So for some people, a simple question about the value, a result of some measurement, could be already highly value laden, because then the question is, okay, who measured it? Whether it was done by the industry or by someone I trust. And in many debates, this dimension is often forgotten, and when it's taken into account, I think it can only contribute to the quality of the debate. So transparency and bringing to the table all stakeholders with their all interests, all the values, and we have to accept that these values may be different. That in many of the areas we will never agree that there is a common set of values, and we will have to find a policy that takes into account this disagreement.
Yes. Can I just a short follow-up question? It is not here, but it's in relating to that topic. How do you think is that best to organize? You have pointed a little bit at an observatory, and how do we organize this best that this happens? Listening to those people, and what role would you see, for example, for the Commission or for the European Union or for other, or for even for researchers and in that?
Yeah.
I think that has to be a diversity of institutional and organizational things. That's not one solution fitting all. But at least one organization being in charge of organizing such public discussions could be helpful because it is visible. Then you have a European Forum for Public Engagement, let's be an agency or an observatory. Let's first start with a European observatory for public engagement, and then this could coordinate different agencies that should be involved, or different institutions that should be involved, and we can see which topic is most important for the people now. And we could have journalists involved and so on. So it could be a forum which is visible because it has a name and it has an organizational framework. And under this umbrella, there could be coordinated a lot of measures and a lot of discussions and different fora.
It's difficult because, I mean, as has been mentioned, each country can have a different way of doing things or different understanding of how you should do this. And so actually the WHO is going to try and survey using their local, regional, country-specific offices to try and gauge what has been done in this area, not necessarily genome editing, but what public engagement activities have happened and how they worked, and try and get some feeling of the best way of doing this. And then hopefully, again, one could try and use that network to help spread how it should be done, and hopefully locally it will be picked up. It has to be done locally, I think, but if it can be coordinated, it would be good.
Yeah, it could be an umbrella with the networking. So in Germany we have opened up the Futurium. That's kind of a museum, but it's a fluid museum. There's an exhibition, there's a laboratory where you can do something, and there are events. And this could be, let's say, organized on a platform, because of course not every European citizen can come to Brussels or Rome or Paris or so, but it can be a platform, and then it can network with specific institutions responsible for that in the countries, in the member states, and then they can organize things in cooperation or alone or whatever. So, but if there's a visible European voice or platform, that would be great.
Fantastic. Thank you very much. That was a very concrete suggestion, I think, and which will be certainly in the report that we also do from that session. I would like to go to one of the other questions there. That has been there permanently there. The difference in risks between spontaneous genetic changes, often unknown, and targeted intentional changes. Is that something you can reply to? You have a view, if that is asked in that general way, or do we need a complicated 3-year-long risk assessment to do
I'll try to give a relatively simple response. So this is the question can be asked like, what is more dangerous, a scalpel or a shotgun? Well, it very much depends who holds it and how is it used. So I don't think a generalization can be really made based on the tool alone. It can be also done meaningfully when the context is provided. So it very much depends. When the concern is about the medical application and trying to make a very precise change, then a scalpel-like tool is more fit for purpose than a shotgun-like tool.
Yeah, I'd like just to add a comment. Of course, spontaneous genetic changes have led to our evolution as a species, and so they're not all bad, right? So I think it's the advantage of these targeted intentional changes is that you can perhaps overcome problems due to a bad genetic change. The issue I was trying to raise about understanding genes and genomes, though, is that we might not always know what is a really good change to make in a gene. So we might think we know. So you may have the ApoE4 allele which increases your risk of getting Alzheimer's, but if you changed it to ApoE3, that might have negative consequences in the context of your genome. So we just don't know enough we're not quite clever enough. For some things, we are clever enough. So I gave the example of sickle cell disease. We know enough about that mutation that we know if we change it back to the normal variant of that gene, then it's going to be okay. But in many other cases, we didn't. And Hayes Janku's crazy experiments in China, he was working with the CCR5 gene, the receptor for HIV, and we just don't know enough about that gene, so his experiments were always going to be crazy.
Thanks. Can I go to or do you want? Sorry, I don't want to cut you off.
I just read two questions.
Yes.
No, absolutely not. And I think they belong together. How much had the negative public perception weight in the regulatory position and so on? And considering genome editing vast potential across multiple sectors, how can we ensure societal acceptance?
Go ahead.
I think that, and I think there are very different perceptions and discussions in different member states. Yeah. So we cannot say the European public has this or that attitude. But I think the only way to change these deep rooting attitudes, because it goes to the fundamentals of our existence, of our conceptions of naturalness and so on, and what the human being is to take the public with the science and the applications from the beginning, having at the local point round tables saying how do we want to shape our environment and our future? Do we want to have this project? And when we have this project, in what kind shall we apply it and shall we shape it? I think the only way to tackle this difficulty is dialogue. Is really talking to each other, and not everyone has to understand in detail whether they are nucleases or other proteins. They just have to understand what is going on and what the effect is and what the risks are, because they're afraid of something. And it's not to talk to them and say, once you understand science, everything is wonderful, and then you accept it. It's not to teach from the bottom, from the top down, and say then if you want to understand it, it's all fine. It's really learning from those who are afraid and to shape together with them where we want to go.
Yes.
So if I can make a point. So I mean, one problem that the EU finds itself in is that it tends to regulate things according to method as opposed to product. So and that's why it's what's happening in, for example, genetically modified crops is different when you compare the US or Canada and the EU. So it's very difficult to use these techniques, even though they're very precise, to genetically alter a plant in the EU and have it marketed, because it's classified still as a genetically modified crop, and it doesn't matter how you do that genetic modification, it all becomes very, very difficult. If you simply focus on the product and say, is this product safe to eat or not, then that's, in a way, a much better way of dealing with things. You can pick a plant from the wild that can kill you if you eat it. You can genetically modify plants to forever, and they'll be safe. So what's important is to regulate the product, not the method.
Yes, and I would like to add to this by relating to what Christine said, that we need a debate that takes into account not only just facts, but also the reasons for different attitudes. Because, I mean, we could speak of the precision of the tool, but it would not help if the actual fear of the public is that who uses it and for what purpose. I mean, we cannot address this by improving the tools. There are other means that have to address the real concerns that the public has, and for this we need to listen to the public for their concerns, and I'm sure that different people in different European countries, different generations, will have very different concerns.
Absolutely, yeah.
I would like to use the remaining... Yes?
They also have hopes.
Yes, of course. Of course.
And probably there are a lot of people out there who want to use the technology to really tackle wide-ranging problems.
Sorry. Thanks. To come back to two of those questions that were touched, there was a question that has disappeared now on the Irish experience with citizens' assemblies. I don't know whether any one of you is familiar, but in Ireland they were selecting for certain questions. I think it was in relation to abortion, for example. They were actually selecting citizens in a random selection procedure to bring them together in a place where then that was discussed, pros and cons and their views, and maybe I'm not explaining this now totally correctly, but it is considered a huge success to reach this way of
So I have been. I know it, and I've been involved in a couple of cases where you have done this, or deliberative democracy, if you like, or where you have these small, they can be small groups of people, individuals chosen at random, and you have, over a period of several days, often to several different occasions, you go through in great detail the issues, presenting them in a neutral way as possible, and that requires a lot of careful thought and oversight generally from opponents as well as people who are proposing a technique. And that can work really well to give you very insightful views into a technology and how it can be applied. We did this for mitochondrial replacement in the UK. Some of the questions coming from the public involved in those things ended up altering the specifics of the regulations that have been adopted. So it's a really valuable...
So it's also a technique, a method to deal with that with that difficulty that was mentioned earlier, that it's actually quite difficult to understand things, but if you have that time to discuss it in this forum, then you can have that can actually even work with people who are not biologists, but you are then able to discuss it.
And even at the level of policymaking, actually.
And even at the level of policymaking, yes. And that leads me to the other... there is this question which has been very often on the top, and I have not asked it to the panel, and the reason is that none of the panelists are representatives of the European Commission. So I actually don't want them to ask to them the question, has the EC reviewed or assessed potential regulatory bottlenecks to the development and commercialization of gene editing technologies for human use in the EU, because none of you is a representative of the European Commission. If you still want to reply to that question, it's fine, but I wouldn't ask you. I would rather take it from a different angle with the regulatory perspective. There has been coming up this also in several questions now. This question of a little bit that relates a little bit to your question with the product versus the technology. I think the current legislation in Europe is actually a mixed one. It's both product and technology, there's this question: novel gene editing techniques potentially offer profound effects for plant breeding to improve crops in Europe. Is not allowed because it is not true that it's not allowed. It can be submitted totally, but it has to respect the GMO legislation in place. And it is true that other countries regulate differently on this. Some of those techniques that are used or some of those gene editing techniques in other countries, Canada, US, are not considered a genetically modified organism according to the law, whereas all of those are considered in Europe. I don't know whether you want to comment on that or whether that is premature or whether that is... I mean, you have your view on the method, yes?
Yeah, I think this question, should CRISPR-Cas be seen as GMO, was answered by the European Court of Justice. But whether this is adequate or not, of course we have to discuss that. But it is genetically modified, yes. Whether it really goes to this directive and how it should be dealt with is a really delicate and difficult and complex question which we cannot address here.
It is genetically modified, but so are plants where you have blasted them with radiation and made many mutations, which are not labeled as genetically modified.
So are plants that just grow in the sun and are radiated by the UV radiation from the sun, and they also get modified.
If you make a precise a specific mutation in one gene, you know what you've got. It's a mutation that could have occurred naturally. Then I personally don't see how that could fall under GMO regulations and in the way that they operate in Europe.
And here I would like to point out that there is a slight confusion in the question, because the CRISPR-Cas is the technique and the GMO is the product.
Yes.
So and this creates confusion very often. That a technique should not be considered a product. I understand that we can regulate on the level of the technique, and that we can regulate on the level of the product, but we have to be very careful to avoid the confusion when we are speaking of the technology, when we mean the product, and we are speaking of the product when we mean the technology.
Yeah, I agree. I can make one comment to you on the more general question. I'm not from the European Union, you see, so I'm not controlling any of the money, but it certainly doesn't help to ban or compromise research. So there are some areas where funding is probably not available, particularly human embryo research from the European Union. And I think that's counterproductive, because if you're not allowed to do research, you will never find out what's possible. So that would be my plea to make sure that research is not compromised. The applications, of course, you have to have those regulated very tightly, and don't just leave it up to scientists like me to say what should be done or not. That has to be society.
Mm-hmm. I think there's only one remark. There was one question about the Irish approach. And there was the sentence, "It is not enough to look at exhibitions in museums." I thought that was relating to my comment.
Yes.
I invite the one to a visit to the Futurium. It's not a classical exhibition in a museum. It's an interactive exhibition. And you just have to inform yourself before judging something or discussing something. You have to understand it before. And thus, an exhibition, an interactive one, can be an approach, but it's not the only one. It's about events, and it's about a laboratory where you can do something by yourself as well. Just
we have. Thank you very much to all three of you. We have come to an end. I know that there are still a lot of questions out there, and I know that some of you feel probably frustrated that it has not been taken up. And I guess you are still around. And if some of you would like to ask the questions now to the panelists, I guess you are still willing to do this. And otherwise, I would like to give them a real round of applause, because I think it was intensive and not easy to reply to all of this in that short time. Thank you very much.